In the second part of the video, we will be discussing about the endoplasmic reticulum, its two types, rough endoplasmic reticulum and smooth endoplasmic reticulum, nucleus, mitochondria, Golgi apparatus, secretory vesicles, peroxisomes, and lysosomes. So let's start with the endoplasmic reticulum. Endoplasmic reticulum is made up of tubular structures called cisternae, and they are flat in the cytoplasm. In between of them is presence of the matrix, which is of watery consistency. They help to process the molecules made by the cells and transfer them to their specific location. Once again, their layer is lipid bilayer, and they have large amount of proteins, as we have discussed in the cell membrane or the plasma membrane. Now, if there are minute particles or minute granules are present on them, which are called the ribosomes, they will be termed as rough endoplasmic reticulum. Here we have the rough endoplasmic reticulum, and if these are not present, these will be termed as smooth endoplasmic reticulum. The ribosomes are a mixture of protein and RNA. They help in the formation of new protein molecules. The smooth endoplasmic reticulum helps in lipid synthesis, for example, phospholipids and cholesterol, which are embedded into its own wall so that the organelles will grow more rapidly. Now, if the cell won't need this much growth of the organelle, what it would do? They would break down in the form of transport vesicles, or it can be also known as endoplasmic reticulum vesicles. Now, after this goes into the Golgi bodies. Here is the Golgi body. These transport vesicles will come to the cis phase of the Golgi apparatus. Now, the Golgi apparatus is made up of 4 or more stacked layers, that can be flat or enclosed. As here, flat or enclosed. In between of them, we have the lumen. These are also termed as cisternae, like in the endoplasmic reticulum. Now, from the processing and everything, the transport vesicles will come out and form the secretory vesicles, the lysosomes, or even cell organelles like mitochondria. Now, we should understand what is lysosomes. The size of the lysosome is 250 to 750 nanometers. Its membrane is once again lipid bilayer. It consists of enzymes called hydrolases. Like mentioned in the diagram itself, we have the hydrolases present inside them, and the membrane we have the lipid bilayer. The nature of these enzymes is to digest everything. So the lysosome can be termed as the intracellular digestive system. The nature of enzyme, digestive. They eat unwanted matter like bacteria, eat also the food particle, and even the damaged structures. They are capable of splitting organic compound into 2 or more parts. For example, here, protein is hydrolyzed to form amino acids, glycogen is hydrolyzed to form glucose, and lipids is hydrolyzed to form fatty acids and glycerol. Next is peroxisome. They are similar to lysosomes, but they differ in 2 different ways. First, they are self-replicative, or they can bud off directly from the smooth endoplasmic reticulum and not the Golgi apparatus. The enzymes they have is oxidase. They combine oxygen and hydrogen to form hydrogen peroxide. Hydrogen peroxide can combine with the catalase, which can further lead to breakdown of alcohol to acetaldehyde. These can be poisonous to the cell. Now comes the secretory vesicles. As the name suggests, they secrete special chemical substance. They are formed from the ERGA system. They are released in the form of storage vesicles. One more fact about the Golgi apparatus is they can form chondroitin sulfate and hyaluronic acid. Now let's discuss about the mitochondria. Mitochondria again is made up of 2 layers, that is lipid bilayer and full of proteins. Since mitochondria is the powerhouse of the cell, it is mostly present like They are most in number present in cardiomyocytes and least in the adipocytes. The size and shape is variable. The size can be from nanometers to micrometers. The shape can be globular or elongated. The most interesting part of the of the mitochondria is it has DNA, so it can self-replicate. That means it has its own DNA, like the cell has its own DNA in the nucleus. Inner membrane and the outer membrane is the division of the membranes of the mitochondria. Inner membrane is further has cristae, that is increasing the surface area of the mitochondria. The cristae has oxidative enzymes, and even the inner membrane has a lot of enzymes present in it. Inside this cristae will be the matrix, and matrix also consists of enzymes, full of enzymes everywhere, since it is the powerhouse of the cell. So these enzymes work together to oxidize, or oxidation of nutrients takes place, which releases energy and forms carbon dioxide and water. This release of energy is further utilized to form high energy substance that is called ATP. So here is the generation of ATP from the mitochondria. That's why the cardiomyocytes needs they work more, hence they need more energy. So they'll get energy from the formation of ATP, that is by the mitochondria. Now let's discuss about the cytoskeleton. Cytoskeleton is a network of fibrillar protein organized into filaments or tubules. It is of three types: the microfilament, the intermediate filament, and the microtubule. The microfilament is present outer membrane of the cytoplasm. Let's see here. Let's suppose this is a cell and this is the cell membrane, the double layer cell membrane. Like I said, this is the cytoplasm, and nearly here will be called as ectoplasm. So they are mostly present in the ectoplasm. For example, we have an actin. Intermediate filament, for example, we have the desmin, the neurofilament, and keratin. They are strong rope-like structures. Hence, as the name suggests, they are supportive and provide strength. The last is the microtubule. Microtubule, for example, we have centrioles, the mitotic spindles. These microtubules are made up of special type of thick filament or stiff filament called the tubulin molecules. They act as a cytoskeleton. Since it helps in mitosis as mitotic spindle, it participates in cell division, and they act as a conveyor belts for transport of vesicles, like transport of vesicles and granules or etc. Now let's discuss about the nucleus. Nucleus is the control center. It sends the message for the cell reproduction, growth, maturation, and even the death of the cell. It is made up of DNA, which is consequently made up of genes. The genes determine which specific proteins will the cell make, and even the intracellular enzymes. All the further works and function of the nucleus will be discussed later in the upcoming videos. Now, the nucleus has a nuclear membrane also, or the nuclear envelope. Here we have the nuclear envelope. That is again bilayer, the outer membrane and the inner membrane. The outer membrane and the space between the outer and inner membrane is continuous to form the endoplasmic reticulum, and further it can be rough or smooth. Here we can see a nucleolus or a nucleoli in plural form. In nuclei, it is made up of RNA and proteins, which is ribosome in turn. There is no any membrane, unlike other structures. The first genes determine specific type of RNA to be formed. Some of the RNA stays in the nucleoli, and most of them will go out in the cytoplasm through nuclear pores. And these will act with enzyme to form other cytoplasmic proteins. So that's all for today. Thank you.